So just to set the stage for you, like the picture you just saw, we have these lush forests that are filled with ohia trees. And then back in 2013, 2014, all of a sudden the trees started to die. And it's never seen before, nothing like this had ever occurred to our trees. So what exactly was this? All we saw, so people within their backyards, or whether it's a scientist, they noticed that there's a rapid wilting of the canopy. So the lush trees that were once beautiful and green suddenly became brown. It wasn't like a traditional death of a tree where the leaves initially are just falling off, the tree looks dead. The main sign of whatever this was going on was that the leaves turned brown, but they stayed attached to the tree. So there was a dead, wilted canopy, which was unlike most deaths that you would see. When the tree was cut into, scientists found this dark staining throughout, if you can see on the right side of the screen, and there's this dark staining which is in the sapwood, which is the fungal pathogen within the tree. And when this was tested, they found out that it was this fungal pathogen, ceratocystis, which causes the tree to die, and the staining within the bark of the tree and within the wood. So it's not one, but two strains which are affecting the ohia trees on the island. There's a more aggressive strain and a least aggressive strain. So for our project, we decided to focus on the more aggressive strain of the fungal pathogen. So it was now named Rapid Ohia Death, or ROD, to symbolize the rapid death of our native ohia trees.
So here you can see a few examples of the symptoms and signs of ROD. As Abby mentioned earlier, a healthy ohia tree has a full vibrant canopy with no wounds or any decay shown. Here in a recent death, we can see, as Abby mentioned earlier, there is wilting leaves, but it's still attached to the branches and such. There is also signs of wounds or any cankers within the trunks and branches, which is how the ROD transfers from tree to tree. And in the tree that has been dead for a few years, we can see bark that is flaking off, as well as branches that have been brittle and show signs of dieback, as well as wood that shows signs of decay due to the fungus in colors of gray, brown, and black. We can also see some pictures of the two different variants that Abby also mentioned earlier. We decided to focus on the ohia wilt instead of the ohia canker due to the aggressiveness it has on the vascular system. And as we can see here, it has a significant coloring within the wood compared to the canker, which only has localized effects.
So this pathogen can spread via airborne or through infected soil. And in order for it to get inside the ohia tree, it needs to go through a wound, whether it's caused by an animal or broken trees or broken branches or even human activity. Even a small wound can provide an entry point for this pathogen to enter. And so once it's inside the the tree, it spreads to the vascular system, specifically the xylem, where that's the part of the tree where it transports the water from the roots into the leaves. And this pathogen basically causes the tree to choke and prevent it from getting enough water to survive. And so this leads to the rapid wilting and the death of the tree. And it doesn't only stop there. These dead trees leave behind dead wood, which then spreads the pathogen even further.
So the fungal pathogen that is causing ROD is known as Ceratocystis leuco-ohia, and unlike traditional nomenclature in Latin, leuco-ohia is a Hawaiian name for this fungal pathogen. So let's just break it up to really understand what the name means. So they're both derived from the Hawaiian words. The first half, luku, means destruction or destroyer. And the second half, ohia, symbolizes the death to our native ohia trees. So as we put that together, we have the destroyer of ohia, or the ohia destroyer. So that is how we got the name, or how scientists got the name Ceratocystis leuco-ohia. And then the other strain, which we didn't focus on, but it's pretty interesting to know, Ceratocystis huli-ohia, which is just a lesser version of this. But the main focus for our project was Ceratocystis leuco-ohia, or the destroyer of ohia.
So we can see here that we used Hawaiian plant and fungal extracts. We used 100 different plant extracts as well as 8 extracts of the marine fungal strain FM324. Here we have a few examples and images of the extracts we used. For example, a hibiscus flower, a Hawaiian gardenia plant, we used the stems, leaves from tea leaves. We used the fruit from beach plants as well as the rhizomes from ginger.
Here are some other examples of the plants that we used. We have black turmeric down here, tea leaves, soursop leaves in the middle, and noni on the bottom right.
So for our experimental processes, all was done in vitro, so in our controlled lab. First, we had to create the inoculum to inoculate our plates. So we took, on the top right picture, is the Ceratopsis leucohea growing on a plate. We flooded the plate, and then we created a 1 × 10⁵ suspension. Using this, we mixed the correct amounts of our flooded leucohea and mixed it with sterile distilled water to create our inoculum. For our in vitro plate testing, we spread the inoculum on the plates and let it for 15 minutes to dry, so it would be on the V8 auger plates. On top of the plates, we placed sterile 6 mm filter paper discs on the surface of the auger plates, and on top of the filter paper discs, there was 7 microliters of natural products were added. So in the plate 1, which is the third photo down on the right side, were some of the examples of the small amounts of the extracts that we placed on the filter paper discs.
So after inoculating plates and applying the extracts, we then wrap the plates in a plastic lab film in order to prevent contamination and reduce moisture loss. We then transfer it to an incubator set at 28 degrees Celsius. This is the ideal condition for leucohea to grow and the best time for us to observe any effects. We then incubate it for about 5 to 7 days, and this lets the fungus grow and also lets any zone of inhibitions to develop. We then mark the north, south, east, and west points of the zone of inhibitions, and this lets us be able to measure with a caliper the diameter of the land, and in order for us to quantify the effect of these extracts.
So the top 2 photos are examples of our controls. So it was the effect of the controls on the inhibition of Ceratopsis leucohea, just to make sure that everything in our processes were correct. On this side, so not this half of the plate, but on this half is our sterile distilled water, and as you can see, there's no zone of inhibition because it doesn't inhibit the growth of Ceratopsis at all. And then for our positive control, we used a 10% bleach solution, which was effective in creating a zone around the filter paper disc, which it was placed on. The bottom here shows the marine fungal extracts, which were all proven to be effective, and each created a zone of inhibition around the filter paper disc that I was placed on. But then what we mostly focused on was here.
We mostly focused on these certain extracts just because of the results they gave. So out of the 100 plant extracts, we got the most positive results from black turmeric, soursop leaves, and kava root. As we can see, the zone of inhibition from the black turmeric had the largest average.
So some of our next steps, because we'd love to apply our findings to help limit the Ceratopsis leucohea, and we're gonna explore the potential as these extracts as an alternative to isopropanol substitutes or tool sterilization alternatives, where there's scientists go out in the field and they want to prune, but there needs to be cleaning materials, which we're looking for that in the future. And then similarly, we can stay away from fungicides because we'd like to limit the introduction of non-native anything really into our forest. So we're going to keep testing to be able to see how we can apply our findings into real-world application. Before we continue, we'd just like to thank all of our mentors, because without their help, it wouldn't be possible within the lab, and our mentors at the University of Hawaii for helping us with the fungal extracts, and then to all of our teachers and for our great audience today. So we thank you for that. And sorry, but the last slide just says thank you for your time, and we'd like to accept any questions at this time. Thank you.
Thank you very much for your insightful presentation and clear explanation. Now we would like to ask some questions from the audience. Does anyone have any questions?
Not with us, but in the very back.
Okay. Good morning, and my question is there any intervention for the problems that you stated with the fungus before? Is there any interventions by by the governments or the locals?
So that's a great question. Currently, that is why we chose to focus on this, because rapid ohia death is so strong that there isn't a way to stop it at all. And it was first discovered in 2014, so you'd think 10 years later and there would have been something, but this is why it is so important, and it's because it is so hard to control. And after all these years, we don't have any true measure to stop the fungus. There's only preventative measures, such as when you enter some of our forests, they'll ask you to scrub your boots, or they'll ask you to please dip your tools in sterilization to try to help prevent things, but there is currently no solution to help kill this fungus. I hope that answered your question. Thank you.